Yeah, so 15 PhD students based at various institutions all over Europe. And this symposium series marks the final stage of the project, where we showcase both highlights from within our own network and also from outside of the network. And each of the ESRs that you see here, they either present a talk or host one session. And today, Nataly, Canales, and I will be your hosts. The series, yeah, so on the next slide, please. Yeah, so this series revolves around 5 themes, which will be presented on the last Thursday of each month. And we're kicking it off today with a session on historical specimens and ancient DNA. And this session is called Biodiversity Time Travel, the use of historical plant genomes in biodiversity research. And we will focus on the use of ancient DNA to study biodiversity changes over time in a range of species covering crops, invasive species, as well as endangered species. So we will have 4 researchers, including 2 almost or recently graduated PhD students. But before I introduce the first speaker, I would like to let you guys know what is the easiest way to interact on Zoom to make this seminar go as smoothly as possible. So on the next slide, thank you. Another one. Awesome. So while the speakers are presenting, please mute yourself. I think we would prefer to keep cameras on so the speakers can actually see who they're talking to, unless there's some bandwidth problem, and then we can think about turning them off, but I hope that will not be necessary. There'll be 5 minutes for questions at the end of each talk. So if you want to ask a question, you will find please raise your hand by clicking on for me, that would be the reactions button, but on this screenshot on the next slide, you will see, I guess it depends on the version of Zoom that you have. So you can either click on participants and raise hand, or on reactions and raise hand. Or if you would, so you could either unmute yourself to ask a question, or write a question in the chat, where we will keep track of what's being asked, and we can also say it out loud for you. Okay, so then our first keynote for today will be Hernán Burbano, who is currently an associate professor at University College London at the Department of Genetics, Evolution and Environment. Hernán studied veterinary medicine and biology at the Universidad Nacional de Colombia in Bogotá. He obtained his PhD from the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where he worked on the ancient genomics of humans and hominins under the supervision of Svante Pääbo and Michael Lachmann. Afterwards, he moved to another Max Planck Institute, this time for developmental biology in Tübingen, where he did a postdoc on the integration of ancient DNA in plant genomics under supervision of Detlef Weigel. He led his own research group on ancient genomics and evolution in Tübingen from 2014 until 2019, when he joined the UCL Center for Life Origin and Evolution. Hernán has pioneered the use of herbarium specimens and archaeological remains for evolutionary plant genomics using standards and techniques similar to those that have been used in human ancient genomics. He studied organisms as diverse as modern humans, hominins, plants, and their associated pathogens using a variety of ancient materials. His research tries to untangle different processes, including the dynamics of past plant pathogen epidemics, the timing of key events in crop domestication and the spread of agriculture, and also the colonization of new ecological niches by invasive or introduced species. We're very honored to have him here today, and Hernán, the screen is yours.
So hello everybody, and thank you very much for that introduction. Let me share my screen. All right, it's a pleasure to be here, and it's a pleasure to see like so many people watching the talk. So it's like over 60 people. So today I have entitled very broadly, and I call my presentation a retrospective view on plant genetic diversity using ancient DNA. And I want to start very general, telling you that I'm an evolutionary geneticist. I'm very interested in evolutionary processes. And this is a very simple cartoon way of depict those processes as the interactions between the phenotype, the genotype, and the environment. And what I would like to argue today that goes in line with the title of the symposium is that to really study evolutionary processes, we need to introduce an arrow of time, because evolution can be understood as a historical process. So a way to introduce this arrow of time is what I call a retrospective approach. And this approach is possible if DNA can be retrieved from ancient or historical specimens. So here in this slide, I present you the type of samples that I have worked with, starting from sediments, animal remains, and when we talk about plants, we have the main sources, plant archaeological remains, and herbaria specimens. And you can see from the time frame from which each type of sample is collected that we have a very wide dynamic range to study different evolutionary processes. Focusing in particularly at the various specimens, for example, we have the advantage that sometimes if we look in detail, we can find samples that have lesions that are compatible with infection of certain pathogens. So we have the fortune to be able to sequence the genome of both plant and pathogens from those specimens. And recently in my lab, we have started to look at insects just to introduce a different player that sometimes also cause biotic threats to plants. So having all these samples and retrieving DNA from them allow us to take this retrospective approach that is just looking directly at the past. Before I move on, I want just to mention shortly that how the DNA that we retrieve from those samples look like. And this DNA is biochemically known as ancient DNA. So the first characteristic is that the DNA degrades with time, so it gets short. Here there is a distribution of a very young herbaria samples of 150 years old, where you see that the median length is about 50 base pairs. In addition, all those projects are metagenomic in nature because only some fraction of the DNA that is retrieved is endogenous, meaning that it belongs to the plant or the animal that you're interested to. And then there is a big component that varies among samples, which is metagenomic in nature, where you have a lot of microbes that could have been present at the time of collection or death, or could have colonized the tissue post-mortem. And finally, when we look directly at this DNA and we align the DNA to a reference genome and ask which type of substitution we encounter, we see that we encounter an excess of C2T substitutions. This is the red line that I'm showing you. So the y-axis is simply showing you the percentage of substitutions that occur along a reed. So on the left-hand side, you have the 5' end, and then it goes inward to the reeds, and you see that we have this excess of substitutions, and we understand biochemically really good what is happening. And this type of characteristics and patterns are normally what we use to validate the samples and be sure that what we are retrieving is historical or ancient in its nature, depend on the timing. Though I'm not going to talk a lot about the technicalities of dealing with ancient DNA, what I would like to argue today is that generating high quality ancient DNA is still very, very important. So in my lab, we have put some effort to contribute to that aim by developing targeted enrichment methods for these type of samples, by developing tests to look for the authenticity of DNA, and also by developing methods to retrieve short DNA. So all of those methods are important to get the best out of all these precious samples that are curated in museums all around the world. I just want to give a shout out to this recent paper that was that summarized some of the methods that we use to retrieve and sequence ancient DNA, and that was led by Sergio Latorre from my group and Rafael Gutaker, which is now research group leader at Kew Gardens. All right, but we gathered here today to talk about plants and plant biodiversity, and we are all very interested in understanding how this beautiful diversity that we see in the world came about. So what I'm going to do during the talk is that probably I could have entitled crop biodiversity. I'm going to talk mainly about crops, and I'm going to talk about plant domestications and the spread of agriculture. In this map, you see very also beautifully represented the centers of origins or diversity of different crops all around the world. And what I'm going to bring your attention is that some of them were domesticated in tropical and subtropical areas, and post-domestication, they cross latitudinal gradients and disperse, for example, to the temperate zones, both at the north and at the south. And this is a process that I'm very interested in. And one of the general questions that we ask in my lab is, how did crops spread and adapt outside of their native range? So that is post-domestication. What I'm going to do today is I'm going to present you two examples of two different crops that they move post-domestication at different times. The first story is about ancient maize adaptation to temperate North America. And that was published already some years ago, and that was led by Kelly Schwartz, which is now group leader at the Gregor Mendel Institute in Vienna, and Rafael Gutaker, which I mentioned is now at Kew Gardens in collaboration with Ed Buckler. So what we're going to look is at flowering time in maize. What we're going to use is flowering time as a proxy for temperate adaptation. And the logic behind that is the following. If you look presently at inbred line of maize in North America, you see that flowering time, which is here showing on the y-axis of this inset, is stratified by latitude. You see that the inbred lines that are at the north of the United States, they need to flower earlier than the ones in the middle or the ones in the south. And the logic is that they need to flower very early in the north, because otherwise it's kind of Game of Thrones problem. Winter is coming, and then the plants would not be able to enter the reproductive stage. So we have variation in flowering time in inbred line in North Americans, and this is stratified along latitudinal gradients. What we know about flowering time in maize, and that's important for this talk, it is what is called a complex trait. So it means that there are many loci of small effects that affect the trait, but the heritability is very high. So it is comparable with human height, where you have a lot of loci with small effects, but also the heritability is over 90% of flowering time. What we know about maize domestication and the introduction of maize to the Southwest is the following. So the map that you're seeing of the America is colored based on a unit that is called growing degree days, which is an accumulation of heat. So the redder the areas, the more heat is accumulating. So you see that the map gets paler when you start to move to the temperate zones. And we know that maize was domesticated about 9,000 years ago in present day Mexico, in Balsas Valley. And then it started to spread all around the world. If we look at 4,000 years ago, it already spread up north, and then agriculture took off immediately. So the dates there at the start of agriculture. So 4,000 years ago, it got as north as I'm showing in the map, and agriculture started immediately. But if you move a bit more up north, even though maize got there 4,000 years ago too, agriculture started 2,000 years later. So agriculture was delayed in the temperate uplands for about 2,000 years. You see that this is already looking paler in the map. And then the hypothesis that we had at the time of why we wanted to investigate that is that flowering time was not fine-tuned to be early enough to be in sync with the environment at the temperate North America. Fortunately, we had samples from an archaeological place called the Turkey Pain Shelter in the Colorado Plateau in present-day Utah. And those were samples excavated already during the 70s. This is a view of the stratigraphical column. So on the top is more close to the surface. And then you go down through the column, you see numbers, which are the layers from which the cups that are shown at the bottom were retrieved. So to make the story short, what we wanted to do is first to date those cups to be sure that they are about 2,000 years old. And indeed, they were very close to 2,000, so 1,900 years before present. And we take an approach where we did whole genome sequencing of those samples. We have different type of samples and the sequencing went really good. And what we wanted to do with the sequences of the maize was to carry out phenotypic predictions. And this is the type of phenotypic predictions that get some time in the newspaper, where you can read that Neanderthal have like red hair, for example, which is a very easy phenotypic prediction because you know the gene that codifies for the color, and then you just look at a particular single nucleotide polymorphism to decide if the color was red or not. So, but the challenge that we had at the time is that we wanted to look at that cobs, and then based on the genomes on those cobs, we would like to say when were they flowering, when they were planted 2000 years ago. So in the next part of the talk, I'm going to show you how we carry out those predictions. So for that, we resort on maize land races. So those are locally adapted cultivars. And this is the distribution of the land races that we look at across the Americas. And those were sequenced for this project. And I'm going to summarize the information about the whole genome data in a multidimensional scaling, where I'm reducing the dimensionality and cluster the samples in that space. So you see how the samples is spread around the PCA space, the color match the colors in the map. But what I would like you to focus is that these pink samples, which are from Turkey pen, are not an outgroup. So the diversity, they are within the diversity of current land races. This is important to remember. So we sequence Turkey pen, and then when we put Turkey pen in the context of modern diversity, it is within modern diversity. So we have these maize land races data sets. What else would we need to carry out the predictions? So we needed the inbred lines. So those are the inbred lines from North America that I introduced at the beginning, where we know that there is diversity in flowering time, and that diversity is stratified according to latitude. So that was the training data set that we used. That was genotype and phenotype before the study. So there is a big advantage of a lot of genetic resources in maize. In this project, we contribute with the sequencing of the land races. And those land races were planted in 3 different areas in North America, in Arizona, North Carolina, and New York. And then they were, in addition to be sequenced, they were phenotyped in 2 different seasons to see at what time they were flowering. The idea then is to hide the phenotype of the land races, build a model only based on the genetic relationships, and do the prediction of the land races using as a training data set, the inbred lines. So by doing that, one can then, since the phenotypes are known, we can ask how well are we doing with the prediction for the land races. So the answer is that we were doing like very good. This is the predicting ability. You have the prediction in the inbred lines and the prediction in the land race parents. And you see that the predictions were really good, though the ability was about 70%. So then the leap that I would ask you to do is that given that we can predict the phenotype of the land races based on the inbred lines with 70% predicting ability, and given that the genetic diversity of the archaeological samples is within the diversity of the land races, we think that that gives us a way to pseudo-validate the prediction that we would perform on the archaeological samples. This is a summary of the flower time predictions. You have a flowering time on the y-axis. You have the different populations on the x-axis. And I'm going to walk you through to the predictions. So this is the prediction of the tropical land races that can afford to flower much later. But then we found that the predictions of the temperate land races, they need to flower earlier. So then the question was, where was Turkey pen located? And then Turkey pen, which is appearing there in pink again, was already within the variation of the temperate land races. So it was already flowering early, 2000 years ago, when we know that agriculture was starting at this particular part of North America. So from that, we can conclude that it is indeed possible to do prediction of previously unknowable complex traits using archaeological samples. And the Turkey pine was early flowering and marginally adapted already to its environment. The next thing that we ask, and I'm going to present here, is that we wanted to know where this adaptation come from. For that, we did this very simple exercise. You have again the map of North America, and then we select that the most temperate adapted land race, which is in Canada, and compare that with the center of origin in Mexico. So we select a set of SNPs that we think are related, that are very different between those samples and that are related to involved in adaptation. And what we're going to ask is like how many of those SNPs were already segregated in maize wild relative teosint. So what I'm showing at the bottom is the distribution of SNPs segregated in teosint that are found in the land races. And what I'm going to show now with a line is from those SNPs that are very different between very adapted temperate land races and the ones in the native range. So they are preferentially segregated in the land, in the wild relative teosint. So from that, what we conclude is that those alleles that are important for temperate adaptation, in the case of maize, were selected from pre-existing variation. So with that I finish, and I'm going to move to the second related part of the talk, which is the origins and adaptation of European potatoes, reconstructed for historical genomes. That was carried out by Rafael while he was in my group too. So instead of looking at flowering time, we're going to look at tuberization. And the story is similar because potato was domesticated in the Andes. And in the Andes, it's only tuberized only short days, these land races from the Andes. So under long days, potato is not going to tuber. That's not a problem in the Andes because you see that the day length is constant along the year. But it's going to be a problem when potato was moved to Europe after the Spanish conquest, because you start to have days that are short, very close to winter. Second part of the story that is important is that we know that potato already moved to the southern temperate zone in prehistoric times, and those potatoes are adapted to shorter days too, which would be the mirror image of what happened in the northern hemisphere. We know that in contrast to maize, where we have a lot of loci involved in flowering time, in potato there is a major player that is called cyclin Doph factor 1. This transcription factor is very important for tuberization onset. There is an active variant that is present in the Andes that doesn't permit the tuberization under long days, but we have allelic variations of this gene that is truncated that permits tuberization. So this is just to show that in transgenic lines where the variants were introduced, this is a 1-1 under long days, the one that is active. It is, if one overexpressed the variant 1.2, it can tuberize under long days. So we wanted to see what happened with potato when it got to Europe. For that, we look at samples starting from the 17th century up to the present. This is how the samples look like, and some of those were collected in the 19th century for then people that became very famous in biology, like Darwin. In addition to that, we team up with the Potato International Center to sequence present-day diversity. This is the samples that we sequence along South America. We didn't do whole genome sequencing, but just capture sequence. And we have about 100 samples with 32 historical samples. So the first look at this data that we look was we assembled the chloroplast genomes from all these samples, and this is how the tree look like. You see that we have a separation between germoplasm from Chile and germoplasm from the Andes. And in the Andes, we could tell apart also if they are diploid or tetraploid. If we look at this data in time, also we have these wild potatoes, Solanum, Demysum, and Vernay, or wild relatives better. If we look at this data through time, we see that at the beginning we have Chilean germoplasm that was subsequently replaced, Andean germoplasm that was subsequently replaced by Chilean one, and at the end we see some chloroplast of the wild relatives. So we harness also the nuclear data and allele frequencies, and we saw that this story was replicated in the nuclear genome. when we saw a decrease on the Andean ancestry at the expense of gaining Chilean ancestry through time. So this analysis and a more formal modeling revealed that the potatoes collected between 1650 and 1750 were closely related to Andean land races. And then after the introduction to Europe, they had mixed with Chilean genotypes. But now you would ask what happened with this gene that was so important, the CTF1. This is just to remind you that we have allelic variation, where the 1.1 native variant is the one that doesn't tuberize under long days, such as in Europe. So what they're going to show you is the ascertainment of the CTF variants in those potatoes. So the plot are going to show you this type of donuts that are at the bottom, where the center show you the type of chloroplast and the surrounding show you which variant is present. If the surrounding is gray, it's going to be the active variant that doesn't tuberize, and any other color is that you have the variants that confer tuberization under long days. This is how it looks in the Americas. So you look at the Andes, you see that they're like mostly gray. So these are the inactive variants. We found that they were not segregated in the Andes. We found some instances dead, but then we found that they were admixed. So from that, we conclude that the inactive variants were not segregated indeed in the Andes. So they were not part of the standing variation. When we look at Chile, which is a very interesting case because we know that there are potatoes that are adapted to long days. We found that the 19th century samples also have only the active variant. And then we found different type of variants in the 20th centuries, but they were unfortunately admixed, so we could not ascertain they as land races. So this is like amazing because it means that those potatoes in Chile, they are adapted in a CTF independent way. So present day Chilean land races were admixed with European cultivars. That was not very informative for our purposes. And only the active variant of CTF1 was present in the 19th century historic and Chilean samples. So they should be adapted in a different way. What happened now in Europe? So if we look at Europe through time, we see that also that at the beginning, only the active variant was present. But then if we move through time, you see that different colors start to appear. So, but since this variant was not segregated in the Andes and also was not found in the few samples that we look in Chile, what we said is that the adaptive variants were detected in Europe during the 19th century, and then might have appeared de novo in Europe. And I put an asterisk because, you know, more sampling in Chile would be beneficial, but with the data that we have, it seems that the de novo appearance of those variants in Europe is reasonable. So with that, I'd like to finish, and I would like to contrast the two examples that I presented. So in the case of maize, I show you data about flowering time as a proxy for temperate adaptation, which is a complex trait. We use archaeological samples, so we travel to 2000 years before present, and we discovered that the adaptation that permit early flowering time come from a standing variation 2000 years ago. Whereas in the case of the potato, we look at other like phenological important point for the plant, which was the onset of tuberization. Whereas in contrast of maize, has a much simpler architecture. One gene plays a major role. We only got back a couple of 100 years to look at what happened based on historical specimens, and found that the adaptation that conferred onset of tuberization under long days probably happened de novo in Europe. So with that, I'd like to finish to acknowledge a lot of people that have contributed to those projects. I already mentioned Rafael and Kelly. If there are postdoctoral researchers listening that would like to join my lab and be included in this list in the future, please contact me for possibilities to write a fellowship together. So those are my funding sources. And with that, I would be happy to take questions. Thank you.
Yeah, thank you, Hernán, for this talk. I think we already had one question in the chat that was posed by Marcel Polling. Marcel, would you like to repeat your question out loud?
Yeah, sure. Yeah, it was only a short question. Thanks for the talk, Hernán. You mentioned at the very beginning of your talk that you used the deamination of DNA to check if it's like old DNA. Could you tell me what you use to do this?
I think that for that particular plot is probably from Map Damage. Yeah, so as you mentioned, so there is a software called Map Damage that uses already reads aligned to a reference genome and stratify the substitutions based on at which position the read occurs, and then you can generate this type of damage plots indeed. Yeah.
Great, thank you.
Thank you.
Okay, if there's any other questions, unfortunately, not all the speakers fit on my screen, so I might actually miss a waved hand or whatever. So maybe
I want to stop sharing so that you can see the people.
Yeah, maybe. And then it's still 3 pages actually. So, but anyone who wants to ask a question, please speak up.
I think
Otherwise,
Barbara has a question.
I have a question. And then people can still.
Barbara
has a question. Okay.
Yeah.
Okay. Can you hear me?
Yes.
Okay. So thanks very much for a very nice talk. I really enjoyed listening to it. And I was wondering, from an evolutionary perspective, you now talked a lot about the plant genomes themselves, but what about pathogenic fungi inhabiting them? I'm thinking about Fusarium, which is always involved in a host plant range competition. Did you pick up those genomes as well? And are you planning to work on these too?
So it is, as I suggest at the beginning of the talk, it is possible to sequence like other microbes present there. That always depends on the sample. And the problem with some of those microbes is that, so the genomes are smaller and the amount of DNA that proportionally correspond to them is lower. So there are 2 ways to do it. Like you can sequence deeper to try to reconstruct them, or you can capture them, just design probes to capture them. In the case of samples, when they are collected at the time of outbreaks, it's easy to go to the lesions and try to sequence the pathogens. So there, we have done work together with people that are also like here in the audience, working on potato late blight, working on Phytophthora infestans, and also, we have looked at the metagenomic content of some of the other samples. So you find the usual suspect living in the phylosphere of leaves. It is challenging. They're challenging in many ways because you want to show that those microorganisms were present at the time of collection and not that colonized the tissue postmortem. And it is challenging because they occur just as a fraction of the total DNA. So one needs other methodologies. I think we are in the lab working on some methods, but also like some people here watching the talk in the audience also have published papers to mine and to use like intelligent strategies to mine microbes and figure out which ones were present at the time of collections and which other colonized the tissue post-mortem because they like to live in herbarium, for example.
Yeah, because I have colleagues in University of Amsterdam, and they do experiments where they try to induce evolution. But of course, this can only happen in very limited time series. So expanding a time series would be, I think, really very interesting.
Indeed, yeah, I totally agree.
Nice. I think if no one else has a question, then we should move on to the next speaker. Nataly?
Can I say something before you move on? So thank you again for the invitation. I'm very thankful to Margret and Nataly that moved my talk at the beginning. And I apologize to the other speakers that I need to leave now. But I wish you like a very enjoyable afternoon.
Thank you so much, Hernán.
Take care. Thanks.
Bye.
Okay. So continuing with our next speaker, the next one is Thibauld Michel. He has a background in molecular biology and peptidomics in the Lille University of Science and Technology. He has worked for the Irish National Research Institute in Agronomy on association mapping and genome-wide assisted selection, also known as GWAS. In energy grass and forage breeding. And he has also worked on agronomic subjects. And with his PhD, he has discovered the domains of botany and genomics applied to taxonomy. He is now fascinated with the idea of using ancient material from herbarium as marker from the past to get information on evolution, on the evolution of a population. He's a PhD student in the Royal Botanic Garden Edinburgh, and his subject is focused on assessing the genetic health of begonia populations using a high-sec approach on living plants from the garden collections and plant samples from the herbarium. Okay, I hope I made some.
Thanks. Yeah, no, that's fine. Thanks, Nataly. Could you see my screen?
Yes, we can see it.
And could you see the pointer, the red pointer?
Yes.
Okay, so so I'm going to start. So today I will talk to you about a side project of my PhD project. We talk about winter flowering begonia or Christmas begonia. And here you can see already a spoiler that the ancestor of the horticultural begonia, that's Begonia socotrana, which has been depicted here the year it has been described in 1881. So first, lately, indoor plant has become increasingly popular for some reason. And looking at the time that the accession indoor plant has been Googled since March 2020, we can see this huge rise of research. So yeah, the lockdown made indoor plant very popular. But actually, they have always been very popular. We can find a trace of indoor plants that were kept in decorated pots in Sumerian civilization, in Egypt, in Asia. And actually, they became even more diverse in the 18th century when botanists brought back in Europe exotic plants from abroad and made the first crosses and developed floral varieties. And they were particularly popular in Victorian era for people of middle class because they were showing the social status of the people that can afford them. And even the architecture has changed by the time, making this huge window and high ceiling to host all these new plants that has been bred lately. And nowadays, they are still popular, as I said. And when we came to consider the surface where we keep these plants, it's quite a huge surface. I think it has been estimated that 1.5 to 6% of the non-ice land on Earth is indoor space. So space to keep some biodiversity. Biodiversity, which is exposed as different selective pressure. It's largely unstudied. And in some cases, some plants that are preserved inside and could be threatened in the outdoor. And so we can keep some biodiversity that maybe don't exist anymore in the wild. So that was basically the start of the project. That was to consider that some diversity we keep, some biodiversity don't exist anymore in the wild. Of all these indoor plants, begonia is a very important one for a long time. And as an example here, we have this flower carpet on the Brussels ground place. And this pattern, this colorful pattern is made of 1 million begonia that are brought together to cover a surface of, I think it's 2,000 square meters. So yes, begonia are very popular. I will just present begonia very quickly. That's a pantropical genus, one of the largest angiosperm genera. There is now over of 2,000 species. When I started my PhD, that was 1,800, so that's growing quickly. And they display a large array of different morphologies. Species are very different. You can see it here in this begonia from section Celocentrum, that's an Asian clade. And we have a very large array of diversity of shapes. And their population structure is very peculiar as well, because they tend to have a very restricted seed dispersal, and consequently that make them prone to be isolated in small population that become inbred over time, that are affected with genetic drift, and therefore a bit more vulnerable. There is this example of a begonia from Madagascar that grow in a single population of 10 to 20 individual, and that's the only of the species. I'm working on two specific species that come from the Socotra Island. So that's an archipelago of islands that is located in the Arabian Sea. And it's quite famous for the biodiversity found over there. I think there is 30%, roughly 30% of the species that are endemic from the main island. Begonia socotrana grow in the Aegir mountain here in the northeast. And Begonia samaensis grow in this little island of Sama in a very, very restricted area, which is, I think it's 1 kilometer or 500 meters. So it's very small. And my first task was to assess their genetic health based on the pattern of homozygosity I could find in their genome. And I was studying it over time using herbaria specimen. So coming back to Begonia socotrana, it has been brought back by Sir Isaac Barley Balfour, who was the regius keeper of the Royal Botanic Garden of Edinburgh by the time, and maintained in a collection called logically the Balfour collection. And it has proved very promising to breed new varieties of begonia, crossing it with South American begonia, tuberous begonia. So there is two groups in the winter flowering begonia. There is the Begonia iemalis. We have 3 examples here. And the Begonia cementia. And Begonia cementia group has an ancestor, the first hybrid that has been made, called the Gloire de Lorraine, that has been made by Lemoine. We have an example here. And so our research question were, so where does they come from? Where does it come from the Socotran parent that has been used to make this cross? Is it several population? Is it the Balfour collection? Is it a single specimen maybe? So we begin to ask this question and to ask, are the alleles that are still existing in the horticultural population still exist in the wild, and is the biodiversity we found in horticultural hybrid is not larger than what exists now in Socotra? So there was a cytological study on the genome of horticultural begonia. So here, genomic in situ hybridization, and that's the genome of an hybrid. So the small chromosome here are in blue are the Begonia socotrana chromosome. The cyan one are from tuberous South American begonia, and there is satellite chromosome here shown with arrow. And it came quite as a surprise that most of the Begonia socotrana genome is still present in the hybrid genome. So to make some more inquiries on that and know the origin of the hybrids, we collected different specimens. So principally the Begonia iemalis, the Eliator one, in different places in the Taipei flower market in Glasgow and on Dutch breeders collection. And in the botanical garden, a recreation of the first hybrid of Crementia, Gloire de Lorraine, has been made. So we sampled this one as well. And there was a sample from unknown origin coming from Texas. So here are the first work that has been made by a student, Philippa Stone, on the genome. And we just realized that most of the variants, the ECNPs that we found we call in the genome of the horticultural hybrid, are actually Socotran variants. There is roughly 75% of the variants that are coming from Socotra, with the exception of the F1, and which has a higher content that could be due at that just because it's a single cross, so there was less admixture in his history, and the Texan hybrid show a very low rate. So obviously there was a lot of introgression of different other sequence in his genome to give this kind of result. So another work of Philippa has been the repeat content within the genome. So on the left here in blue, there is the wild type genome, including Begonia socotrana, the ancestor. And on the right here, the hybrid repeats. And as you can see, the hybrids are on average have a higher number of repeats in their genome. And that could be explained by the genome shock that happened during hybridization. There is a burst of transposon activity and sequence replicates. And so that's why we can find this higher level of repeats. Once again, no, not once again, we got this surprise with the Baladin one here from Biekenkamp, and we just think that this sample has been contaminated. That could explain the lower level of repeats. And on the F1 that show a level, the Gloire de Lorraine hybrids that show a level comparable to Begonia socotrana. And once again, it's just because it's a single cross. So there was less hybridization in the history of this sample. And we notice that most of these repeats are made of a common long terminal retrotransposon. It's TY3 gypsy, which is quite common among plants. And so we explore the repeats of this retrotransposon in the genome of the hybrid. And here we have an output of Repeat Explorer. On the cluster of dots, you can see each dot is represent a read from the data set. And the closer the dots are, the more similar are the reads. And so on the pattern of dot, the cluster of the dot we obtain, we output, we can see all these different shapes, which are very different from one another. And that could indicate that there is a lot of diversity in the genome structure of the hybrid. And so that they had a very different breeding history. At that point, I started to build my pipeline to analyze the genome of Begonia socotrana and samaensis. So I collected different specimens from different location and time points because I was going to analyze the evolution of genetic health over time with a time series of samples. And I was particularly interested to analyze the genome of the hybrid because we have the original Begonia socotrana specimen from the Balfour collection that has served to make all these hybrids. So basically, the project was to sequence it and to compare the pattern of allele between this sample and sample from different other population to the hybrid we have sampled. A very little word on the technique. We have used the HypeSeq method, so target capture method. The bait has been designed by Catherine Kidner, based on functional sequence and conserved sequence. And so we have sampled different capsule on herbaria specimen. There is some example of the material we have gathered here. And here is a very rough figure of my pipeline. First, we use for the alignment, we use pileomics. We incorporate map damage that we were talking about earlier to basically avoid the bias due to ancient DNA in herbarium specimen. Called variant, we have to segregate the duplicated sequence, so the paralogs and the orthologs in the genome. And for that, we use different tools, and we compare the result to a mapping population we made with South American begonia. And after that, we set up a series of scripts to phase the variant and to discriminate, basically to deal with the polyploid horticultural specimens that are tetraploid. So once we have the variants, we have the Socotran one and the horticultural one, we can filter the horticultural one with a consensus of the Socotran variant to filter them and output the Socotran specific variant in the horticultural hybrid and relate the pattern of allele we will find with the one we will find in the historical specimen from different population. Here are just preliminary results to show you the success of the target capture with the IPSIC method. Unfortunately, we got a few reads with the most ancient specimen, with the blue being the total read for a sample, and the yellow the success of the target capture. So it's really, really low for the historical specimen, but we got quite a lot for a specimen who were 30 years old, and a good coverage as well. So we at least we can use these ones that are referenced, that has been correctly annotated, and so we have their population of sampling. Here is as well some studies I made on the SNP, the number of variants we found in the genome of the horticultural hybrids here in blue, and the match, the intersection between this variant and the Socotran variant from two populations, Scand and Dixam. And strangely, I found far less variants than Philippa did. She found something like 75%, and I found only 5% match between Scand and 0.8 with Dixam. So I think that could be explained because I'm a bit more stringent because I'm selecting only the Socotran sample on the bait set we are using. So it may explain this result. And I will compare the pattern to know if we have been stringent enough. And eventually, we are just about to output some results. In the next few weeks, I'm working with one student to finish the pipeline and eventually have the genetic history of the horticultural hybrids. So we will, first thing we will do is to make some population genomics on the genetics on the different populations that grow on the Aegir mountain. To study as well their homozygosity level and pattern to know if they are in good genetic health or not. And after that, relate them to the horticultural hybrids to know what population and maybe what specimen has been used to make the crosses. And if we don't find any match, we can ask the question, is the allelic diversity we found in the horticultural specimen still exist in the wild, or has the population disappeared over time? And so in this case, the horticultural hybrid would be the last remnant of this allele diversity. So here is my team. I'm working with Zitian Tank for the moment, but I have to thank all my team and of course Plant.ID for helping me in this work. And thank to you for your attention.
Thanks for such great presentation, Thibauld. I was wondering if there are some questions now.
Hey, Thibauld. Good job. interesting study about the repeats. I was wondering, what is the genome size that you're working with in these begonias?
It's very variable between different accession of the horticultural hybrids.
So when you have something like 50% of the genome was repeats, in what range of genome size should I think?
I'm sorry, what?
I thought in the graph you had something like 50% of the genome was like repetitive elements.
No, I think that was not percentage. Wait a minute.
Sorry, maybe I misread the graph.
It was a coverage for
okay.
50x, I think.
No, that was in the analyzed cluster. Sorry, that was in little on the top left of the slide.
Okay, let me ask the question like this then. Do you know what proportion of the genome
is made of repeats? No, I don't know exactly. That just considering the different cluster, but I don't know exactly how many repeats we have.
Okay.
If we have other questions, maybe we can type it on the chat, but otherwise it's time for a break now. We have 5 minutes break. Margret, please, I'm asking you.
That sounds good. Yeah.
All right.
I agree.
Let's stretch our legs.
Yeah. Okay. See you back in 5 minutes.
See you in 5.
Was that 5 minutes? Roughly, okay. Yeah, people slowly coming back. Okay, I'm wondering if we should wait for more people to turn their cameras back on, or if we can just assume that they're there.
No, I think let's wait a couple of minutes more.
1 more minute?
Mm.
Or maybe we can start at 10 past instead of 5 past? Okay. Looks nice and cozy in the cabin.
I think 1 more minute and we're ready to go. Most people are back.
One more minute for people to hide their kids. No, I'm joking. Kids are welcome in this seminar too. Okay, should we start again? Yeah, I think so. Okay, so our next speaker is Vanessa Bieker, who is currently wrapping up her PhD at the Norwegian University Science and Technology under supervision of Mike Martin, who is also here, I think. Vanessa has always been deeply fascinated with nature and history, and throughout her studies, she has been working to combine these two passions. She first studied chemical biology at the Technical University in Dortmund, Germany, and she followed this up with the Master's in Biological Anthropology at the Johannes Gutenberg University in Mainz. And for her master's thesis, she used ancient human DNA to explore the spread of the lactase persistence gene in Europe. And for her PhD in evolutionary genetics at the NTNU University Museum in Trondheim, Norway, she switched from humans to plants. Now Vanessa uses historic herbarium samples to look at the genetic changes over time in the invasive weed Ambrosia artemisifolia in both its native and the introduced range. She recently handed in her dissertation, which has been approved for defense. So congratulations to Vanessa. And after her defense, which will take place on the 19th of March, Vanessa would like to continue working on plant genomics, and I'm sure her future will have many exciting things in store for her. Vanessa, we're honored to have you here today, and yeah, feel free to start sharing.
Thank you for the introduction. So let's see. Can you see my screen? So yeah, I'm talking about using historical herbarium specimens to look at evolutionary genomics of plant invasion. So first, like, what are invasive species? So invasive species are species that get introduced to a new location, mostly through humans, and through global trade, more and more species get introduced. And there can be a threat to global biodiversity because they might outcompete local species there and drive them either to local or global extinction. And if you take one species away from like the species interaction, it might be that this whole tower of species interaction like falls apart. But invasive species are also really nice to study evolution, because you can kind of look at each introduction as an experiment that goes on for many generations already, and quite more than we can actually look at when we now just start an experiment. And invasive species, they need to overcome certain barriers. So first it's geography, so they first need to actually be transported to the new location. And once they get introduced, they also need to be able to survive in that new location and establish a population and actually reproduce. So this is the stage where they actually become naturalized in the new habitat. And if they start to spread to new environments, you normally call them then invasive, and they can have then an impact on the ecology of the new place. In those different stages of invasion, there are different traits a plant or a species needs to have to establish or become invasive. So at first, to establish, you might, like selfing might help, because you don't need that many individuals then to start a population. But other traits might become more important in other stages. And if you actually now want to look at the genomics of this invasion, if you just use modern samples, you only look at the snapshot, and you only look at one of the stages. But with herbarium specimens, we can actually look at this continuum, or look at different stages of the invasion process. So this is where herbarium samples come in. And there are quite many herbarium specimens in the world, and many of them are actually also digitalized. And they go back to like the 1600s, but most samples are actually from the 19th century or later. So it's like the last 100-200 years, we actually have quite many specimens that we can use. And my study specimen is common ragweed, or Ambrosia artemisifolia. It's a weed, and it's quite widespread, and it can be relatively small, but also quite tall. Here you see me next to a plant that's taller than me, which, well, I'm not that tall, so it doesn't take that much, but yeah, it can be up to 2 meters. And you may have heard from it. If you have hay fever, you're quite likely allergic to Ambrosia as well, because it's been pollinating and has like super allergic pollen. That's why people usually want to get rid of it. And also it can grow within crop fields and then reduce the crop yield, especially like soya beans, or it's closely related to sunflower. So if it grows in sunflower fields, it can also reduce the yield a lot. It's native to North America here, where it's really pretty widespread. But it got introduced to all continents now except Antarctica. So it's really common in Europe. And that's where I concentrate on the native range and on Europe. And in Europe, it's mostly like in the south, because it can't, it's an annual weed, so it can't up in Norway, or here where I live now, it can't fulfill the life cycle. So it dies with the first frost, so it dies before it can actually produce seeds. But with global warming, it's quite likely it can expand to like further north. And it's also in the European range, where it got introduced in the late 19th century. with contaminated horse fodder, and today the biggest source of introduction is with bird seeds. So seeds are then introduced to Europe. And yeah, in the native or in the introduced range, the plants are actually larger and they show better reproduction. And there's also a latitudinal decline for flowering time and reproductive biomass, which is similar to the decline observed in the native range. So it indicates that there has been rapid adaptation in Europe within a quite short amount of time. So I'm using almost 300 historic herbarium specimens and 350 modern samples to compare them with, from both the native range and the introduced European range. And I do use low depth shotgun sequencing, and we have a reference genome. So I could align those reads to a reference genome, and we ended up with coverage of a mean coverage of 1.3x for the historic samples and 2.9x for the modern samples. And because that's not that high, and we actually weren't aiming for higher because we wanted to include more samples, we used genotype likelihood methods instead of actually calling SNPs. And we wanted to see from where in the native range the European samples or the European populations actually came from, and if there is population structure in the introduced range already, and also if there has been adaptation. So when we look at the population structure, it's like there are no really clear clusters. It's probably because this plant is wind pollinating and the pollen can travel quite fast. So there's like, except for the south here, almost no samples are fully assigned to one genetic cluster. But there are still like 3 main clusters in the native range. So like 1 south cluster, 1 east cluster, and 1 west cluster. And the structure stays the same over time. So it's the same structure in the historic samples as in the modern samples. Oh, I forgot to mention, basically all the historic samples were collected before 1940. So they're like from around the time they actually, the plant got introduced to Europe. And there's also between those 3 extremes or 3 main clusters, there's this one admixed cluster. Also in both ranges, it already exists in historic times, and it was proposed that maybe admixture can lead to more invasive success of a species, because then you have like more genetic variance. And also some groups might be pre-adapted because they're, or populations might be pre-adapted because they live in similar environments as they're found now in the invasive range. And also this admixed population, it's mostly a mix of west and the east cluster, and the south cluster, it doesn't really contribute that much to this admixed population. And the European samples, they're here. They and now those black spots on the PCA, they look more like the admixed cluster, or some I actually look quite similar to the west or the east cluster, but basically none of them look like the south cluster. So the south didn't really contribute to the invasion in Europe. And also the west, this light pink here, it doesn't completely disappear in Europe, but it's less prevalent in modern times. And also in modern times, no sample is fully assigned to that cluster, whereas in historic times, there are quite a few that are fully assigned to it. And in the native range, the structure stayed the same, but actually in Europe, it didn't really. There are those populations here that actually become their own or get their own genetic cluster. So there was a lot of change over time in these regions. And there are also those outlier groups here in the PCA. Those are, and also here, those are those samples that are fully assigned to a cluster. So despite there was very little time since the plant got introduced, there was already quite a lot of change. It maybe because the population size was quite low, so it was a lot of drift could happen. And also interestingly, those clusters here, they're not found at all in the native range. So it's something completely new to Europe. So it's quite likely it actually occurred in Europe. And then we looked at selection in Europe. So if some genes or regions are actually under selection there, because we saw those big differences. And we used FST outlier analysis for this. And we actually found that their selection on or against defense against pathogens, drought resistance, plant growth, floral morphology, and flowering time. And this is kind of in line with phenotypic differences that were found in European populations. So their European populations are less drought resistant than native North American populations. And the European ones show increased growth and higher reproduction. So that's with floral morphology and also flowering time. And the defense against pathogens. So we're not sure if it's positive or negative selection, but it's in line with the evolution of increased competitive ability hypothesis. So that's the hypothesis as to why species might be so successful in the invasive range. And it says like, because they're actually escape from enemies in the native range, so maybe herbivores or pathogens, they're able to allocate resources away from defense mechanisms towards increased growth and reproduction. And since we found evidence of selection on all these traits, it's quite a good indication that this hypothesis is true for common ragweed in Europe, which is pretty cool, I think. So yeah, in summary, we found that there was an admixed population or cluster that was found in the native range already before the plant got introduced to Europe. And most of those European populations or samples actually look like this admixed cluster. So the admixture in the native range might have helped this plant to actually become so successful in Europe. And we find drastic changes in the population structure in the invasive range, but not in the native range, indicating that there was actually quite a lot of selection and adaptation within a short time period. And we also found evidence of selection on traits that are in line with this hypothesis as to why invasive species might be so successful. So yeah, this I want to thank like all the people who helped in this project, and especially like all the herbaria for providing all these samples. Yeah, and thank you for actually having me here today. Oh, well, having.
Thank you, Vanessa. Does anyone have any questions? Barbara again. Sure, go ahead.
Sorry.
Yeah.
Can you hear me? Thanks very much. Very interesting presentation again, lots of data. Again, you mainly talk about the plants, and we also published work on invasive species. But there we discovered that the native food web actually adapts quite rapidly. So now these plants are winners in their non-native range. But do you expect this to last for a very long time?
No, I think it, well, in the invasive range, the pathogens and herbivores co-evolve with the plants. So I think it, well, it depends on how fast those enemies can actually adapt to the new plant. But I don't think it will take that long for it to not be, yeah, for the enemies to kind of win over.
Yeah, they have multiple generations per time, so they reproduce much faster than the host plant. Thank you.
Cool. Then there was a question in the chat by Marcos. Marcos, would you like to ask your question out loud, or should I repeat it from the chat? I'll just repeat it here. Okay, he is asking what were the most challenging aspects in aDNA extraction?
Well, actually, the extraction went quite well. So we didn't really had many problems with the extraction, but there were, like, in general, the concentration is quite low. And so because the concentration is so low, they're quite prone to contamination. So we worked in clean lab facilities. So yeah.
Okay, I hope that answers your question, Marcos. Great, he says, thank you. Is there anyone else who'd like to ask a question to Vanessa? I don't see any hands raised, so if not, then I'd like to, oh wait, okay. Tiziana is asking, how is flowering time calculated?
Flowering time, yeah, it's like in Arabidopsis, you have genes that are associated with flowering time. So we used, we basically mapped our genes to the Arabidopsis ones to figure out which functions they actually have. So then we actually have genes with the known function of flowering time in Arabidopsis. So it might be that they actually have different functions in different plants, but yeah, that's the closest you can get.
Okay, thanks again, Vanessa. And I think we'll move on to the last speaker, which Nataly will introduce.
Okay, I'm very happy to present Jasmin Ramos Madrigal now. She has a background in genomic sciences from Universidad Nacional Autónoma de México. She moved to Copenhagen to do a PhD with Tom Gilbert at University of Copenhagen. And there she focused on the analysis of sequencing data derived from archaeological plant samples to study their domestication processes. Since then, her work has mainly focused on using ancient DNA to study the domestication of maize, grapes, and dogs. She has also worked in paleoproteomics to study the evolutionary relationship of great apes. Currently, she is a postdoc at the Center for Evolutionary Hologenomics, where she keeps studying plants domestication, but now from a hologenomic perspective. And the floor is yours, Jasmin.
Thanks, Nataly. Thanks for the introduction. So I'm going to share my screen now. So could you please tell me if you can see my screen?
Mm-hmm. Yes, we can see perfectly.
Check. Perfect. And can you see also my
yes, we can see your cursor.
The cursor. Thank you. Thanks. Okay, yes, thanks for the invitation to this symposium. I'm very happy to be here, and I'm very happy to show you some of the work that we have been doing on studying grapes from a phylogenomic perspective. So yes. Okay. Give me 1 second. For some reason, I cannot change slides. Okay. Okay, sorry, there we go. I was having a little bit of problems. Okay, so I'm going to start with a little bit about grape domestication in general. So grapevine, the grapevine has been with us humans for a long time now. It is believed to have been one of the first fruit crops to have been domesticated. It was domesticated somewhere in this area that is between the Black Sea and Iran, and sometime around 8,000 years ago, and perhaps much earlier. And well, with most of the archaeological and genetic data agree that it was domesticated somewhere in this area, but it's also quite debated. And after it was domesticated, humans spread it through the world. It moved with some of the main civilizations of that time, like the Romans and the Phoenicians and the Greeks. And today is one of the most important fruits worldwide. We consume it as fruits, and we use it to produce wine, for example. And of course, with domestication, there was several changes that happened both morphologically and genomically to the grapes. For instance, if we compare the domesticated grapes with its wild counterpart, we will see that the morphology of the leaves, of the seeds, and the size of the fruit is different. Also, while the wild grapes are dioecious, the domesticated grapes are hermaphrodite. And well, today we have a huge diversity of domesticated grapes, which mainly are categorized in these two groups: wine grapes, which we see here in this part of the slide, and then we also have table grapes, which we consume mostly as fruits. And something that happened also with domestication, something that changed, is the way we cultivate them. We cultivate grapes. While the wild grapes, they reproduce through seeds, domesticated grapes, we cultivate them vegetatively, which means that we don't use the seeds, but we use plant cuttings. And this also means that, for example, in this field, we can have the same plant cultivated multiple times, and all of these plants are genetically identical. Another consequence of this is that, for example, if we were to buy a bottle of wine of Zinfandel from France, a bottle of wine of the same variety from Australia or from Mexico, they are all going to be the same, produced with the same variety, which are identical clones. So this concept of grape variety usually is synonymous of an identical clone. And also another consequence of this is that, well, in 2010, in this paper, it was found that if you compare the genetics of different varieties of grapes, we will find that they are highly related. So there are a lot of first and second degree relationships between all of the most important grape varieties today, which we can see here in this network, for example. And of course, as I mentioned before, grapes have been with us for a very long time. There are records of Romans using grapes to produce wine. We have paintings, for example, also from ancient Egypt, of people using grapes of different colors to produce wine, for example. But one of the main questions in general in grape domestication and in viticulture is how is it that the grapes from these ancient civilizations relate to the varieties that we use today? When is it that we started proliferating these grapes vegetatively? Also, for how long have we been cultivating these same varieties, right? Because we can see from the pictures, for example, from the Egypt, that they use red grapes. But how does those relate to the red grapes that we use today? And well, more general, could it be that some of the varieties that we consume today, that we use to produce wine, for example, were selected for people in these ancient civilizations, and if maybe we still use them today? So these were some of the questions that we were trying to study, and of course, using, in this case, ancient DNA or phylogenomics, and using this idea that if we look at these ancient samples, that will help us to directly look into the past. So today I wanted to tell you about these two studies that I worked in one of them during my PhD some time ago, and another one that is more recent, where we use this idea of trying to identify ancient seeds that are related to modern grapevine varieties. So for the first study, we studied 28 archaeological grape seeds that were collected from different parts in France, mostly from southern France, but also some from the north. And they were mostly from the Roman period, but we also had one from the Iron Age and a few from the medieval period. And then we also had some samples for this second study from Georgia. So in this case, we had access to 17 grape seeds that were from these different ecological sites in Georgia. And they were also from different periods, but mostly from very recently. So approximately 500 years old. And so what we did with these seeds was extract their DNA, build some Illumina libraries, and in particular, we enriched for approximately 10,000 SNP sites, which we know are informative to identify all of this modern diversity. And then we sequenced them. So here in this picture in the right, you can see Anne-Catherine, who was the one extracting some of the French seeds. And something that always amazes me is the size of the grape seeds. As you can see here, they are super small. So yeah, but we still were able to extract DNA and build these libraries. And well, from the French seeds, of the 28 seeds that we studied, 21 had good coverage. 7 were not so good local, but still decent coverage. But from the, oh well, yes. So from the Georgian seeds, only 3 of them had a good amount of endogenous DNA. So DNA that comes from the actual seed and not from the microorganisms that are in the sample. So yeah, we have 1 good seed and 2 others that were somehow decent. And so we compared these 10,000 SNPs on these ancient samples with a genotypes panel that contains this diversity of modern cultivars, approximately 800 modern varieties from all Europe, across Europe, some wild grapes as well, mainly from Europe, but also representing different areas, and that were sequenced as well in these 10,000 SNPs that we know are, again, informative for differentiating between these varieties. So that if we build an MDS, like the one that Hernán was showing in his presentation, here we have points which represents different samples. We have here all of the varieties, the approximately 1,000 varieties. We can differentiate between wild grapes, which separate in one side, domesticated grapes, which separate in the other. And we can also see different clusters of, like regional clusters. For example, West Europe here in purple, we have table grapes here in orange, and these other 2 clusters, right? So we compare first the seeds with this data set in using this multidimensional scaling approach. And what we found was, well, first, that all of them fall in the domesticated side of the plot, right? So all of them are seeds that derive from domesticated grapes, which kind of makes sense because, well, they were found in archaeological sites and they are not so old, but still it confirms that the people that were consuming or using these grapes, these grapes were actually cultivating them and not just gathering from the wild. And so if we zoom in into the domesticated part of the plot, so we exclude wild grapes, what we found was that all of the French seeds, which are these black circles, they actually fall within the diversity of modern varieties from West Europe. So from the same area where they are from, right? While the Georgian seeds fall within the diversity of East Europe. So again, within the diversity of modern Georgian grapes. So this was very cool because first it told us that there is some kind of continuity. So the grapes that were used in the past in those regions are still used today or are still cultivated in those same regions, both in France and in Georgia. Yeah, so that was quite cool. But then the next question that we had was, okay, we know they are domesticated. We know that they are similar to the grapes that were cultivated in this, that are today cultivated in these semi-rich regions. But is there any direct relationship between the ancient seeds and the modern varieties? So are there any clones, let's say, that have been cultivated since then? So to try to answer this, what we did was to use these identity by descent patterns, which is basically this sites. So we count sites in the genome where a pair of samples, a given pair of samples shares 0, 1, or 2 alleles that are identical by descent, so that they were shared through their parents, right? So for instance, here in this green rectangle, if we have two individuals which are identical twins, for example, or in this case, the same variety, identical clones, we would expect them to share exactly 2 alleles identical by descent across all sides of their genome. So this is what we did. So we tested pairs of samples in the first case, 2 pairs of ancient samples to see if there were any pairs that had this kind of relationship. Okay, and what we found was, first, among the Georgian seeds, we didn't find any of this kind of relationship, which was okay. But then among the French seeds, we found several of these relationships. In fact, we found 19 clusters of seeds that were identical across their genomes. So here they are represented in the map, where each circle represents a different seed, and the colors represent varieties. For example, in the case of these purple clusters, we have in this site, we have 3 seeds, 2 of them from the same variety, which are identical clones, and another one from a different variety. And something that we found very interesting is that there was one variety that was shared by 2 sites, one in northern France and one in southern France. And this same variety was identical. So meaning that between these 2 sites, either they were transporting the same varieties, but also potentially they were cultivating them vegetatively. So this is kind of evidence that since the Roman times, the people were already cultivating the grapes vegetatively, which matches with archaeological and historical records. Okay, but then this was only taking into account pairs of ancient seeds. So the next question that we had was, is there any link between the ancient seeds and the modern varieties? So now we look for these same kind of patterns, but now with pairs of ancient and modern varieties. And what we found was, first among the Georgian seeds, we found one case of one seed that was approximately 500 years old, which we think might be an identical clone of a Georgian variety that is called Adreli scalcana, which probably you don't pronounce that way, but something like that. So the sad thing about this seed is that the coverage was not as good. So we are not completely sure that is identical. We only had like approximately 10% of the sites on the panel to compare, but it seems to be extremely similar. But then we also found another example of a French seed that was 900 years old. And in this case, we had good coverage. And this seed was identical to a variety that is still used today to produce wine, which is called Sauvignon. Yeah, and so in these two cases, we found a link between a modern and an ancient variety. So I have to say the first time that I saw both of these results, to me it was very surprising, because what this suggests is that we have been cultivating or propagating this same variety clonally for at least almost 1,000 years, right? So for me, it was very surprising. Then I learned that from the people that know about ecology of grapes and about viticulture in general, that this is kind of expected, because there is a lot of interest in keeping the grapes homogeneous, so that you can, when you produce wine, this is with the expected characteristics. But still, so this example of French seed, ancient French seed being identical to a modern variety, and the same for one from Georgia, it was very cool, at least to me. Yes. So the last observation that I wanted to show you is that so if we build a network like the one I showed you before, where we have these connections between seeds, and we include the ancient seeds from France, which is from this area that we have more. So if we build this network and we then look at how these connections appear through time, so what we see is that the one seed we have from the late Iron Age is not connected to anything. But when we move to the Roman period, like half of the seeds are already connected with modern varieties, which are here in this case in white. And when we move to more recent times, so to the medieval period, we see that all of the ancient seeds are connected, right? So there seems to be this pattern of as you move to the present, then you're going to find more connections, right? But then the question, well, unless we have this late medieval, of course, which is this Sauvignon, which is super connected and is identical to a modern variety. But then, so we have this question of how far back in time can we go, and we can still find these connections, right? So it's something that we're still trying to answer. So just to conclude, so what we found with these studies is that, well, there were multiple varieties cultivated in ancient Rome, but also both in Georgia and in France. That well, there is this geographic continuity where the ancient seeds are more similar to the varieties that are cultivated today in those same areas. We find evidence of vegetative propagation that dates back to the Roman era. And we find these two examples of identical clones that are cultivated since the, well, almost 1,000 years in the past. So are we drinking wine that was selected for us by ancient civilizations? In some cases, yes. And how many of the varieties that we have today can be traced to past civilizations? We still don't know, but that's something that we would like to know. And with that, yes, so thanks to all of the collaborators. Of course, this is a work of a lot of people. To the organizers of the symposium, thanks for inviting me today. And thanks to all of you for listening. Yes.
Thanks to you, Jasmin. Yes. Are there any questions to the speaker now?
Yes, Catherine.
Hiya. I was wondering if you could do a similar analysis of the genetic variation in your ancient specimens as to what we've heard about this afternoon done on maize and on ragweed, and infer what the characteristics of those ancient varieties were like.
Yes, that's a very good question. So something particular about grapes is that they are propagated vegetatively. So that's why we can do these kind of networks. My guess is that it should be possible with maize, equally wheat, but it's going to be much more complicated because, yes, for example, maize, well, it's propagated sexually. So each generation, yeah, creates like new connections, and then it has a lot of introgression because it is propagated like sexually. It has a lot of introgression from wild relatives. So yeah, my guess is it will be interesting, but it will be much more complicated.
Thanks.
Yes, Barbara.
Thank you very much. Very interesting public presentation. I really like that you found these clones that are at least 900 years old. And this brings me to my question. Do you think it would be possible with those SNPs that you did sequence to also look for somatic mutations and apply a sort of an age estimate?
That's a very, very cool question. That's something that we really wanted to do. I think because we only have 10,000 SNPs, so it's a very limited data set. So in this case, the amount of differences, let's say, that we found between these two clones, because they are not like 100% identical, they are like 99.9 something identical. So the number of differences that we found were consistent with what we would expect from sequencing errors or from just a variation in the analysis. So there was not a huge difference, but my guess is if we were to sequence the whole genomes, then we should be able to do that. The problem is that since we need to extract the DNA from the whole seed in order to have enough, because it's very small, for those seeds that we already sequenced, there's no more sample left. So there's no much more we can do. But in the future, it's definitely something that's, yeah, it's going to be interesting.
Thank you very much.
Could I ask a related question, or is that not allowed for the host?
Go ahead.
I wanted to ask something very similar. Because I was wondering, because these clones are so incredibly old, how often do you think that a useful cross happens and that a new variety generated? It maybe seems to happen very seldomly. Do you have any ideas about this? Yeah. I suppose the varieties or the clones that you find in the archaeological, like you're probably limited by the archaeological record, right?
Yes, so that's also a question that we are interested in. Definitely. So how often is that you create new varieties, no? Either intentionally or just by chance. I mean, today it's like directed efforts, as far as I know. So people directly select varieties and create new varieties, starting from the ones that they know and trying to predict which characteristics they're going to obtain in the new ones. Like in the past, that's something that, yeah, it's definitely something that's very interesting, but I don't know how often. Yeah, I guess for that we will need like a much larger data set to trying to figure out like how often people were creating new varieties. Or yeah, that's an interesting question.
Thanks.
There is one question posted in the chat. How did you deal with DNA degradation? Was it an issue?
Yes, yes, definitely. So in the case of the French seeds, they were actually very well preserved. So they were waterlogged, which apparently is a very good condition for DNA. So those ones were very well preserved. They were degraded. The DNA was degraded, so we have very short fragments, let's say, but that was still very usable, let's say. It was possible to map and everything. It had these patterns of DNA damage that, for example, Hernán also showed in the previous talk. So that was something that we had to consider. For instance, we were very careful when mapping to, for instance, remove reads that map to multiple places or that had a low quality when mapping to the genome, and these kind of things that you do when you have this degraded DNA. And then in other cases, like the Georgian seeds, for example, in most cases, they had no much endogenous DNA, so there was nothing we could do. But I guess that's the case with many ancient samples. I don't know if that answered the question. Okay. And if anybody else has questions, then you feel free to email me or, yeah.
Could I ask one more question? It says no one else. But I don't see any hands, and Nataly is not. So I was looking at this PCA where you have the Western grape varieties and you have the Eastern grape varieties, and I noticed that the Georgian varieties are very close to table grapes. And so I was wondering if table grapes are those grapes that are used for food instead of for wine production, and what could this say about the different traditions of viticulture in these two locations?
Yes, that's a great question, and I should know, but I actually don't remember right now. So yes, there's definitely a connection like that. So yeah, table grapes are a different group, not only because of the use, but also phylogenetic evolutionary. So they were spread by different civilizations. I don't remember right now which one, but they were spread by a different civilization, and they were also like domesticated to be like larger. The fruit was larger, so that it's easier to consume, and they're not used to produce wine. So I'm sure there is some kind of connection between the table grapes and, for example, the Eastern diversity. But also something important is that well, all archaeology and genetics points to grapes being domesticated. So the origin of domesticated grapes is in this area in the East, right? So yeah, so that's what I can say about this connection. But I'm sure there is a connection between them.
Okay, so if we don't have more questions for now, probably we could thank to our speakers. Thanks Jasmin, Vanessa, Thibauld, and Hernán, who has left, unfortunately. And thanks to everyone that has joined us now. I hope this seminar has been very fruitful for you, and we can keep contact with the speakers or anyone here. I've seen there's some interactions on the chat too, so that's very, very, very cool. And yeah, I hope you have a good day today. Thanks a lot. And see you next month for the next series of the seminar. All right, take care. Bye-bye. Maybe a last picture, right? Maybe, yes, I will make one now. Okay, say hi or bye. All right, perfect, thanks.